Good morning, everybody. My name is Ameen Eetemadi. I'm from the Tagkopoulos group. Okay. I'm from the Tagkopoulos group from UC Davis. So the outline of this talk, I'm going to talk about some of our recent work that is published about prediction in the context of multi-omics. And then the rest of the talk would be about continuing work that is a specially designed neural network for this task. So the goal of our lab, one of the main goals, is to be able to predict the cellular state and its behavior given the environmental setting and genetic background. So what's the value of this? This enables us to, once you can do a prediction for a cellular system, it can help lower the cost of your experimental setting. So for example, in synthetic biology, this is very, very helpful. When you have some engineered products and you want to maximize, for example, generation of a given protein, that is, for example, you want to extract drugs from the protein, this becomes very helpful instead of you running a lot of experiments in the lab. If you can do things in silico, this is going to be very valuable. Also, if you want to analyze the effect of different antibiotics, this is going to be very helpful. And so this work consists of a lot of data sets in different levels. So here you can see we have gathered data from different levels. I need to move to be able to see it myself. Okay, so we gathered all data we can get with respect to the bacteria E. coli. It comes from various public data sets. It comes from different layers. You can see proteomics, you can see metabolomics, fluxomics, proteome, and transcriptome. So all these different layers that are involved in the cellular behavior are all integrated, all we could get from the literature and some from our own lab generated. So we gathered a very big data set. And next we try to do prediction of the cellular behavior from all these different layers. So it starts from the input layer where you have different stresses. It can be, for example, heat is a given stress. You would use the genetic setting of the given species, in this case E. coli. You'll see which genes are available, which genes are knocked out, for example, and what is the medium in which this bacteria is living. You characterize it. What are the characterization of it? Once you have this as your input, these are the things that are going to define the behavior of your bacteria in this case. Then we build multiple layered models. The first layer is the transcription layer, where these connections are the connection between different genes, whether one gene is activating the other gene or if it's inhibiting the other gene. We've gathered all that data from the literature and try to see, given the absence or presence of a given gene, how this is going to impact the other parts of the network. In this work, this was done using a recurrent neural network. So once you have modeled your transcription, you can then know, this can inform you about the concentration of your proteins as well, other than the mRNA level of the genes. So if you also can build a proteomics level prediction, then this can also inform you about other parts of this the cellular system. So you can see that this is very interconnected, although different layers have their own system, but we also connect these subsystems together to be able to do a good prediction. Now this involves a lot of data processing, a lot of normalizations of the acquired data, and in this we used the blue water system pretty heavily. Anything that you change in this pipeline, you need to run all the things again. So this is really helpful to have access to this system. Now, what I was destined to do here was to have a very close look at how we design a predictive transcription layer for prediction of mRNA level concentrations. This is in all the system in E. coli. This is the part of the network, this is the part of this system for which we have most amount of data. For the rest of the system, we have data here and there. For transcriptomics, this is where you can bet on if you can do something. Let's start here. So next I'm going to talk about how we approach this. So basically we take this problem as we have a transcription regulatory network information available to us. We have some knowledge about the thermodynamics of the transcription, and we also have transcription profiles that we have acquired from mRNA sequencing, for example. So you have a network, for example, you have a network like this. How should we go about integrating all this knowledge together to be able to have better prediction? So I like to use this analogy that you have an archaeological manuscript for which you have some information is lost. If you want to be able to understand this manuscript, you first need to understand the alphabet that is used in the manuscript. Then you understand how this alphabet is going to make words, how it's going to make sentences. Once you understand that, then that can help you understand what's in the manuscript. So use this analogy here, and this is the basic building block, if you wish, the letters used in the manuscript. And this is the most fundamental unit of the neural network that we designed. In this, you have a given unit has transcription of its concentration of the transcription factors as its input, and we have a decision function that's depending on the gene perturbation. This is going to either be enabled or disabled. And as a final result, this is going to predict the mRNA concentration. So this is the basic building block. As the example that we have right now, that's as a specific function that we use that is working well, relatively well, is we have such activation function, and for the decision function, it is a simple multiplication. basically. If the gene is absent, then it is going to be multiplied by 0, and this is going to work with the backpropagation algorithm, where if something is multiplied by 0, it's not the output is going to be 0, and nothing is going to be backpropagated for training this given module for the given gene. So once we have modeled, once we know our basic building block, then we ought to understand what are the components we build from this, and those are basically genes. So you can have different type of relationships in this basic building block, where you can have a self-regulator, you can have a single transcription factor, you can have multiple transcription factors, and these I call our words. And then when you put these together, you can make small circuits, and each of these circuits is actually in our implementation is a neural network, where the output of one unit is connected to the output of the other, and it works as a transcription factor in our case. So here are basically some of the most common circuits that you see in a gene regulatory network. So we built our network, and you can imagine you can connect these components together and build bigger and bigger networks. But there is some issues when you want to train this network. Basically, by that I mean the parameters of this network. Let me go back. So here you have a bunch of parameters that you need to train. And basically, backpropagation is very sensitive to initial values. If you work with neural networks, depending on the initial values, basically it's going to find a local optimum related to those initial values. We took this strategy that we have two steps basically. First step is you train the individual modules separately. So you take each gene, you know that the transcription the concentration of the transcription factor, you know the concentration of the mRNA level of the product. Therefore, you can train individual modules, and we use the customized linear programming based method. And then once you train each individual module, you have good initial values for the parameters of that module. And then you can train the full network together to make sure you adjust your weights appropriately as a full network. And specifically, we use full batch and conjugate gradient for the final training. So here's our math. Basically, this is where the algorithm which trains the specific modules. It turns out that in the function that I showed you before, this is the function that in this example it has two transcription factors as an input, and it's going to predict the y, which is the concentration level of that given gene. So our goal is to find the parameters that fits well with that data, right? It turns out that among all of those parameters, if you know P1 and P2, if you know two of them, the rest of them you can get through a linear programming, a constraint linear programming. Therefore, our algorithm works as this. We set only two of the parameters, and then we solve this to get the minimum. We get W star. Then we append W star with our initial value. And then for the rest, we use a simple gradient-based optimization, which you just take the gradient of your loss function, which is this, and then you move forward, basically. So next, I'll talk about our data sets. So initially, I started with the whole big real data set, and that data set was very hard to work with because there's so many things going on, so many things that we were unsure of. As an example, the actual network. There's some connections in the network that are known to be well established. Some of them are we don't have as much confidence in those connections. Therefore, we use as an initial point, we use synthetic data. So we extracted different candidate networks from Dream4 Challenge, which is for a network inference in gene regulatory networks. And we used networks for the number of genes range from 3 to 9 in this study. This is very ongoing study. And we use thermodynamic simulation to generate data, so we perform gene knockouts, for example. We do perturbations of the parameters of the network, the affinity of the transcription factors, and we use different noise levels to generate different multiple data sets. And for validation, we simply use mean squared error and 5-fold cross validation. And we, as the initial point, we compare it with various common neural network architectures. In this case, we use feed-forward neural networks. And initial results give us some confidence that this is a good approach, where when we compare amongst all of the data sets, when you compare any given method with the best method on that data set, we perform better than the common architectures. And in conclusion, I would like to remind or summarize as TRNN, which is Transcription Regulation Neural Network, is a deep learning-based framework for this prediction of steady-state mRNA level. It does require specialized training, though. So the usual ways in which you go about optimizing neural networks did not work. I spent a lot of time finding ways to find the parameters, basically. And our next step, key step, is to try to use this on real data and see how it performs. And for scaling up, we're going to use parallel computing provided by Blue Waters in this case. And we'd like to thank the NSF PRAC Award and Blue Waters for funding us. Thank you very much.